Hello, my name is Amanda Crocker. I am a graduate student in Dr. Amita Segal's lab and lead author on our current Neuron paper. Today I'm going to tell you about our recent work on understanding the neural circuitry underlying sleep in Drosophila. This work was done in collaboration with Dr. Erwin Levitan and his postdoc, Mohammed Shahidullah. As most people know, sleep is a necessary and fundamental biological process. We know that a sleep-like state spans from worms to humans and most species in between, including flies. For instance, on the right, we have a bottle of sleep-deprived flies that are now rebounding, and on the left, a bottle of normal, awake flies. What we are focused on is understanding why and how flies sleep, in hopes that it provides insight into why you and I sleep. In this study, we are looking at the neural circuitry underlying sleep in the fly. In mammals, many of the neurochemical centers of the brain have been mapped, with a basic understanding of how they are wired together. This is not the case for the fly. More importantly, in the fly, we have many tools to probe the intricate workings of these circuits, which will be described in this paper. These tools are not currently available in mammalian systems, so understanding how a neurotransmitter system is specifically wired and functions in flies may provide great insight into how it works in mammals. The neurotransmitter system we chose is the octopamine pathway. Octopamine is the norepinephrine homologue in flies. Here I am showing a picture of all the neurons in green that produce octopamine in the fly brain. We have previously shown that when all of these neurons are active, the flies have a dramatic loss, about 70% of their nighttime sleep. In this study, we asked if specific cells producing octopamine were responsible for sleep-wake regulation. Using a technique known as Markham, we selectively expressed a bacterial sodium channel to depolarize subgroups of cells. We found that a group known as the Asm Neurons are responsible for octopamine's wake-promoting action. We then went on to ask where these Asm neurons send their signal. We expressed a molecule that blocks the effects of octopamine, the regulatory subunit pKa, within different neuronal populations in the fly brain, some of which are shown here. We found that an area known as the pars intracerebralis, PI for short, is required for the effects of octopamine on sleep-wake. More specifically, these are the insulin-producing neurons of the fly. We go on to show that the Asm neurons likely signal to the PI. We then asked what intracellular effects occur when octopamine binds its receptor in the PI. We found that the octopamine receptor known as the OAMB receptor is responsible for the effects of octopamine on sleep-wake activity. Expressing this receptor in only the insulin-producing neurons of the PI was sufficient for normal sleep. In addition, bath application of octopamine on these insulin-producing neurons causes an increase in cyclic AMP, as monitored by using a special cyclic AMP reporter. We then went on to ask what effect this rise in cyclic AMP has on the insulin-producing neurons. And we found that application of octopamine causes a decrease in the outward potassium current in vivo, and in theory leading to a prolonged action potentials within these neurons. In our study, we have shown that the Asm neurons, shown in purple, signal to the PI neurons in green to relay their wake-promoting actions. We argue that the PI is homologous to the mammalian hypothalamus because it also has sleep-promoting neurons, shown previously. In addition, it receives input from the circadian system and lateral neurons, shown in blue. The PI is also the major output of the fly brain to the motor areas and the metabolic centers of the fly. In light of our findings, it is worth taking a second look at the role sleep and metabolism play on each other. Thank you, and enjoy.